Okay, I welcome you all to this award talk. I'm Stefan Klump from the Division of Biological Physics, and it's my great pleasure to introduce Erwin Frey, who was awarded the Max Planck Medal yesterday here. And I can tell you, in the Division of Biological Physics, we are very excited about this award, not only because Erwin is one of the giants in our field, but also because Erwin is one of the founders of our Fachverband. So we're very happy, and Erwin has over the years made many important contributions to our field in many different topics, from semi-flexible polymers, molecular motors, to biological self-assembly, pattern formation, and many more things. And we'll hear some of that now, and I don't want to go into Erwin's time much more. So please join me in welcoming Erwin and congratulating him once again.
It's a great pleasure to give this presentation in front of all of you. I'm very honored by that, and I hope I can entertain you a bit about the research that I've been interested over the years. Actually, I picked something which is not an overview of all the research I've been doing. It's the research I've been doing over the last few years with my students that I would like to focus on. I picked a fairly broad title because I think that one man had a vision many, many years ago, which is Erwin Schrödinger, and he wrote this quite famous book on what is life, where he's asking whether one could explain all the beautiful phenomena that you see in biology with the laws of physics and of chemistry. And we have come a long way since that book. We are not there yet in answering all the questions he would have liked us to answer, but we went a long way, and we had made a lot of progress over the last years. What I would like to focus on is two particular topics. One is on the self-assembly of complex structures in biology, where I would like to ask a question which is very similar to a question people ask in computer science, namely, what is the time complexity of an assembly process? And the second part of my talk will be about protein patterns in cells, and those systems really have become some prominence lately because it has been possible to study these protein patterns in detail, not just in the living system, but also in a Petri dish using isolated components. And there I'll try to show you how self-organization takes place in these protein patterns. So let me start. Self-assembly for me is an algorithmic problem. It's a computational problem, and you'll see why in a minute. So if you look at these beautiful structures of self-assembly in nature, from microtubules to flagellar motors, or in the lower row you see viruses or ring-like structures that people have made out of DNA bricks, putting them together in these type of structures that you see. Now, when you look at these structures, then you can ask a thermodynamic question. You could ask how in the free energy landscape the system would reach a steady state, an equilibrium state given by these structures. But that's not what I want to ask. I want to ask, how long does it take from the initial structure to the final structure? And that is a question that is not only one when you talk about a non-equilibrium system. Even in an equilibrium system, this is a different question from the question of thermodynamics, because you have the question of time. How long does it take?
for the system to self-assemble.
let me compare computer science and self-assembly. If you talk about computer science, you talk about algorithms, about a criterion after which you terminate. The time in terms of CPU units is how much costly such a program is. And that depends in general on the size of your program, so the input size. In self-assembly, you have a very similar type of logic. You want to understand the assembly structure as a function of the scenario you pick, so the design you pick. You want to know after which time you have a sufficient yield, say 90% of the yield. And you would like to know how much does that take in real time, and how would that time depend on the complexity of the structure, that means on the size of the target structure that you're trying to build. And that's what I mean with the time complexity. How does time of assembly depend on the structure size S? The way you approach this is, of course, depending on what you're trained in. And I'm trained as a theoretical physicist, so I wanted to understand this in a conceptual, simple model, as simple as possible, but not too simple. So here you see different kinds of building blocks, squares, triangles, hexagons, and they assemble along different pathways. So let's look at that. You have your monomers. They dimerize into dimer structure, and the dimer elongates with the elongation rate nu. Things can also disassemble and go back. So you have a detachment rate that depends on the binding energy that you have. And quite importantly, the detachment rate depends on how many neighbors you attach to. If you just attach to one neighbor, you pay one binding energy. If you attach to two neighbors, you pay exponentially twice that energy. Well, and then it's a matter of limits on how you can simplify the model that you want to study. And what we want to do here is we want to analyze what happens in the limit that you do actually have strong binding. That means it's much, much harder to unbind a building block that's bound to two neighbors as opposed to a building block that's only bound with one neighbor. And that, if you look at it, already gives you a hierarchy of structures. There will be structures which are kind of magic structures that have, where all building blocks have two neighbors, and they're particularly stable in this assembly pathway, and you can construct a theory that exploits the fact that you go through these magic structures one by one. And as you compare these two different types of building blocks, you notice that the nucleation size is different for building blocks which are squares versus triangles. Sigma here is the nucleation size that characterizes the structure. There's other numbers which are important. The other number that's important I call the assembly order, which is gamma, and that's characterizing how much of the building blocks you need to build on the secondary level once you have a nucleus. How many building blocks do you need in order to get the next magic structure that has the same stability as the nucleus that you started with? And then finally, there's a third number, which I call omega, that has to do with how long the periphery is of your building block in order to assemble the structure. So there's just three numbers that characterize the self-assembly process of these structures. Now, when you analyze that in terms of a stochastic simulation algorithm called the Gillespie algorithm, you can tune various parameters, say the detachment rate, and ask what's the assembly time to get 90% yield. And that's what you see here. That's the assembly time to get 90% yield as a function of that detachment rate that I showed. Well, you should notice two things. One is that as you look at different morphologies of the building blocks, the assembly time is very, very different, right? You see here, this is for triangles, that's for squares, that's for hexagons, and they differ by orders of magnitude in their assembly time. There's another feature which is very key here, and that feature is that you need to fine-tune in order to get the optimal assembly time. So you see that here, you need to tune towards that minimum in each of the cases to get the best possible self-assembly time as a function of this detachment rate. Well, now let's say that we have done this, we have fine-tuned our control parameter, the detachment rate, to that minimum, and now ask the question that I posed: what's the assembly time as a function of structure size? Well, here it is. That's what you find. The assembly time as a function of structure size is a power law, and that power law that you get depends on the morphology of the building blocks. This is striking. You see here a power law, and that means as you change the structure size, you get a huge amplification of the assembly time. And as you see, since the powers are quite strongly dependent on the morphology of the building blocks, it makes a lot of a difference how you build your structures, whether from triangles, squares, or from hexagons. This here is another feature of the system. It's this fine-tuning of the detachment rate, and it also turns out that this fine-tuning parameter is a function of the structure size, and it also follows a power law. Well, why are there power laws in these self-assembly scenarios? This is not a critical phenomenon. Far from it. There's no diverging length scale in that system that you could talk about. Well, let's see how we can actually understand that using theory. And with theory now, I mean we have this assembly scenario with monomers, a nucleus, the growth of the existing clusters, and then you can write down an equation of motion, which is just a kinetic equation. And that looks ugly, but it's actually very simple to understand. You have monomers, you lose them for the formation of the nucleus, you lose them for the formation of larger clusters. You have the nucleus, which is gained by this construction from the monomers, and you again lose towards higher order structures. And the last line is just the balance of smaller structures and larger structures as you grow the final self-assembled structure. So these equations you can simulate, and we did, but there's a point. If you look at those equations, they have a symmetry. And that's obvious, this symmetry, when you write them in a continuum form, where you disregard the discreteness of the process, you just replace them by a continuous field theory. And then that field theory looks like so. It's an equation for the monomers, a boundary conditions between the clusters and the monomers, and a growth equation for the clusters, which is basically a drift equation towards the final steady state. Point I want to make is that equation that you see here shows scale invariance. And you can see that by just making an ansatz, a scale invariance ansatz, like so, where you say the monomer concentration or the cluster concentration, which is a function of time, detachment rate, and structure size, actually is only a function, a scaling function of scaling variables that you have here on the right-hand side. And once you realize that this kinetic theory has scale invariance, you're done. Because now you don't even have to calculate the scaling functions, you have to just use the fact that the theory shows scale invariance in order to infer from that the power laws that you saw in the numerical simulations. And in particular, the assembly time, so the yield time, which is a function of detachment rate and structure size, shows this power law behavior with the two exponents, theta and phi, that are introduced earlier. And you see this quite clearly when you look at the yield curves as a function of the detachment rate. You can rescale them nicely onto a single scaling or master curve. The exponents are this. The exponents for the time complexity, and that's the one I want to focus on, is given by this variable theta, which depends, as I told you, just on the three numbers that I've introduced: the size of the nucleus, the attachment order, and properties that are related to the boundary of your self-assembled structure. Is that theory correct? Well, you always have to test against data or experiments or simulations to see whether that's true. So let's do that. Here is the same graph as I showed you earlier. You see that the power laws that we deduce from the simpler theory are quite close. They're not identical, but they're quite close to the numerical simulations that we have found. So from that, we understand that underlying the self-assembly process, there is a scale invariance in time, and that scale invariance in time can be exploited to understand the exponents characterizing the time complexity of the self-assembly scenario. Can we do better? Well, I told you, right, there was this fine-tuning problem. You had to adjust that detachment rate to this particular value, which was a minimum in this curve, and that's not very convenient for a biological system, since biological systems need to be robust to parameter changes in order to fulfill their biological function. So what Florian, who was the student, the PhD student working on this idea he had, was to say, well, maybe it helps to build these structures in a hierarchical way, to start with triangles that have particular strong binding sites that they first form a hexamer, and once they form a hexamer, you then assembly from the hexamers into higher order structures. And indeed, when you do that, what Florian found is that instead of the fine-tuning problem that we have here, the actual assembly time is pretty much independent of that detachment rate. And that gives you a very, very strong independence on that rate. And moreover, you see there's even a reduction of the assembly time. So you kind of hit two flies at the same time. You solve the fine-tuning problem and you improve the assembly time by quite a bit, namely by orders of magnitude. So what's the punchline? What do we know about self-assembly dynamics? Well, I try to convey to you important message that we can think about self-assembly processes in the same way as you can think about computational problems, namely in terms of their time complexity, and characterize them by power laws and scaling laws. And what matters here is something which is unusual for somebody trained in statistical physics, right? The morphology of the building blocks actually matters. So details are important for the long scale, the long time behavior. There's a lot of things to still research in this problem. I've not talked about any spatial or temporal order in the system. That's a frontier. I've not talked about any kind of assembly errors and their control. That's certainly an important question if you want to make structures with a high fidelity, not just high yield. And of course, you can drive the system, you can make it open and drive it far from equilibrium. Now let me turn to the second part of my talk, which I find as fascinating as the self-assembly process, namely how do proteins organize in space and time. And here, as you can see from these two movies, on the left-hand side, you see an E. coli cell, just 2 microns in diameter, that shows these oscillations back and forth between the 2 cell poles, and that serves a function. The function is that this defines the midplane of the cell such that the cell faithfully can divide into 2 equal daughter cells. On the right-hand side, you see a vesicle, just a vesicle, not really a living cell. That's just a lipid bilayer membrane. And that lipid bilayer membrane is moving on a pattern. And that pattern here is this green labyrinth foam-like structure, which is the same kind of proteins that give rise to the min oscillations in the in vivo system. So what's amazing there is that you can create cell-like motion just with protein patterns, no cytoskeleton, no molecular motors, just proteins and lipids. So let me dive into this a bit. The biochemical network underlying the MIN patterns in E. coli cells are just two proteins, MIN-E and MIN-D, and they follow a very simple reaction kinetics, which is explained here. So you have two proteins, MIN-D and MIN-E. This MIN-D protein, importantly, is a protein that's a ATPase, so it means it's fueled such that the whole cycle that you see here, the biochemical cycle, is not a detailed balance cycle. It's a cycle that's driven off equilibrium by the supply of the fuel, which is the ATP here. So what goes on is the MIN-D in its inactive state is activated. Once it's activated, only in this activated state can it bind to the membrane. When it's bound to the membrane, then you have the same type of protein being recruited to the membrane by a nonlinear process. So you have more and more of them accumulating on the membrane. And now there's an antagonist, the MIN-E protein, which is also recruited to the membrane, but now does the opposite. The MIN-E protein hydrolyzes the MIN-D, so it switches it from the active state to the inactive state, and that then makes both of the proteins fall off from the membrane, and you have this whole cyclic process of attachment and detachment from the membrane into the cytosol and back. Now, when you look at this, you can ask the simple question, which people have been asking for drums, right? Can you hear the shape of the drum? You can ask here, can you see the shape of a E. coli cell? And indeed, you can from the patterns, the experiments from Kees Dekker's lab. You see as you deform the E. coli cell into different shaped containers, the oscillations change from pole to side oscillations. What's even more beautiful is these pioneering experiments by Martin Lohse, Fischer-Friedrich, and Kruse and Schwille, and they have put the MIN proteins outside of the living system onto a in vitro dish here, and there you can study then the patterns in a well-defined way, and you see these beautiful spiral waves on a very distinct length scale. Now, can we model that? Well, you can. You can just use the diffusion reaction equations, put them into a finite element code, and you see the same kind of oscillations that you also see in the living system. The main point that we understood at the time, I did this with Jakob in his work, was there's one very simple but strict rule to get patterns. And the strict rule from the model equations was that the MIN-E concentration needed to be less than the MIN-D concentration, simply because otherwise the antagonist would counteract the MIN-D so much on the membrane that it couldn't even establish a pattern in the first place. So you need the antagonist to be weaker than the protein that shows self-recruitment. Well, as a good theoretician, you ask whether that's true. Is the model true? Is it correct? Can you compare it with data? And that's what we did in a collaboration with Petra Schwille's group. That was the work of Simon Kretschmer when he was a PhD student at that time. And what do you see? Well, the MIN-E to MIN-D ratio for which you see patterns goes from 0.05 to 15. So bad, right? This is a contradiction to what I just told you. The MIN-E concentration can be much larger than the MIN-D concentration, and the system even shows patterns. So the biological system shows a much, much higher robustness as what we anticipated by trying to write down these models. And Jonas, at the time, he from reading literature on structural biology, found out that there is another biochemical reaction which is very important in these systems, which is not obvious because it's a fast switch of the protein which are called MIN-E. So what happens there is this MIN-E can open and close. And as it opens and closes, it can only form a bound state with the MIN-D protein when it's open. And the MIN-D proteins opens that MIN-E such that it can bind to the membrane. And this switch is going fast. It's on a sub-second time scale. So when Jonas put this in, he could reproduce the robustness of these patterns. So from that model, you can find that these systems really show the robustness as indicated in the experiment. Well, then the real test is, what if one could switch off this Min-E switch? Well, Simon was able to do that, and once he turned off this opening and closing of the Min-E proteins, you indeed recover the results of the original theory, which has the much lesser robustness, and the Min-E concentration needs to be less than Min-D to show these beautiful patterns. Over the years, and that was very recent, just in the last 2 years, we worked with Suk-Yoon Yoon from UCSD in the US. And Suk-Yoon Yoon, with a student, they actually tried to check our prediction, not just in vitro, but actually in the living system, by creating the biochemistry, which I don't really fully understand, to actually upregulate and downregulate the different types of Min-E and Min-D proteins. So they made a phase diagram of the patterns as a function of the Min-D concentration and as a function of the Min-E concentration. Well, and that was our prediction. You should have a much larger range of patterns. The patterns should get very robust. And indeed, to make a long story short, I could talk a lot about this. There is a nice agreement between the experiment and the simulation. Both show this extremely broad regime of pattern formation, and in particular, the living system, the wild type, is right here on the bottom of this curve, and that means it's optimizing for the production of proteins, which is really a limiting factor in cell biology. It's costly to make these proteins. And it's a bit farther away from the boundary than actually needed, because you need robustness. If the parameters fluctuate a bit, you don't want the system to become unstable. And there's more to it. You can compare the wavelengths, you can compare the time scales, and that also agrees very nicely with the experimental data. So in short, over the many years, we have a full quantitative model explaining the biological process. Well, and then you can ask yourself a question. After explaining all these beautiful biological phenomena, is there something in the game for physics? Is there some new non-equilibrium physics that actually appears here? And my answer is, in short, yes, and let me explain a bit of what I mean by that. So here you see a cell, a C. elegans zygote, where you have a formation of a zone of proteins on one side and the other side of your zygote, such that you have cell polarity. Well, how does that zone, that bipolarity, emerge? In a computer simulation, you see the interface that you see is formed. But it's formed in a particular way. When you look at the length of the interface, it's minimizing the length of the interface. And then you wonder, what kind of phenomena is underlying that? Interface length minimization, you usually connect with surface tension. But there is no obvious surface tension here. This is a system that's driven off equilibrium. It's just a diffusion reaction system. So how can you have surface tension? Well, the question, the answer to that question is, if you look at the interface of those patterns, these interfaces are maintained by the cyclic currents that I have been talking about. The Min-D going to the membrane, going back and forth, and going back and forth. And these cyclic fluxes maintain the interface by balancing the diffusive fluxes that you have in the cytosol and on the membrane, and the reactive fluxes that exchange the particles, the proteins, between the two compartments, the cytosol and the membrane. And once you have that, and you look at an interface at the boundary, There's a very simple explanation why there needs to be a surface tension. As you deform the boundary, you stretch the outer surface and you compress the inner surface. So you change the area outside versus the area inside. And that's different at different parts of this undulating interface. Here you increase the detachment zone, and here you increase the attachment zone. And that imbalance creates a cytosolic particle surplus on the side where you have a enlarged detachment zone, because more particles will detach here in this part of the system and less on that part. Well, and then you have a diffusive gradient. Diffusive gradients try to relax, so we'll have particle transport from that side of the system to that side of the system, and that ultimately flattens out the interface. And the law you infer from that is just a generalized Gibbs-Thompson relation, where you find that the change in the cytosolic concentration is the area surplus that you get, and this area surplus scales with the interface width. That's the analog of the surface tension, and it scales with a curvature. That's a very general law, and the important aspect is really that this here scales with the curvature of the interface. And now when you look at data again, this is again data from Petraschwili's lab. Glock was the student that did the work at this time. You see that these min patterns in the right parameter regime, they actually show structures that are super reminiscent of foams. Foams are very well characterized in equilibrium, and they have features like Plateau laws, Neumann laws, von Neumann laws, and in particular, what's known about foams is that they have only 3 vertices, and the intersection angles are all 120 degrees. And that's indeed true here. In the experimental data, you see clearly that the interface angles are 120 degrees. The number of edges are on average 6. And there's something different here as compared to foams. What's different here is that this system does not coarsen. Foam coarsens, as you know, when you drink beer. Sometime the foam is gone. Here it's not, because there's an antagonistic reaction that maintains the interfaces at a certain structure. So we tested our now full quantitative theory that we have developed over the years against the data. And what you see here is that this complete system, supplemented by an additional reaction on the membrane, which I call a persistent membrane binding, gives you simulation results with the same kind of characteristics as you find in experimental data. You have the 120 degrees angles, and you find that there's a histogram with the edges that average around 6. There's something special about this 6th. Namely, why is it that most likely you get here faces, edges, which have a number of 6? The reason behind is a geometric reason. So here's a computer experiment where you have an original structure and you scale it down to a smaller size. So you want to know what makes the system now regrow to the steady state. And when I run the simulations, you should see that there's a distinct difference between the bubbles that you see that have 5, 6, or 7 vertices. Distinct difference is that the 5s shrink, the 6 stay constants, and the 7s grow. So in that you can then relate to theory in a very robust and interesting way, because as I told you, the velocities here are all scaling like the curvature. Once the velocities change like the curvature, the area changes just the boundary integral over that velocity, so over the curvature, right? And here you have an integral of the Gaussian curvature over a boundary, which we know to evaluate because that's just basic differential geometry, the Gauss-Bonnet theorem that tells you that this right-hand side must be proportional to the number of edges that you have minus 6. So it means once you have figured out the cause of the flow being proportional to curvature, differential geometry tells you that you must have this Gauss-Bonnet law. And you see this as well here from the simulation data. It really confirms the analysis in a nice way. So we do have a law which is very similar to the law of foams, which is based on geometry. There must be more to that, right? Because I told you there's not just coarsening, as this picture shows you, where you have von Neumann's law and some kind of forward topological transitions. There's also anti-coarsening, and that's due to the fact that if you look at these different structures, there's different ways of these domains to split. So here, in the center of the bubble, a new domain is forming, and the domain then forms an interface between the two bubbles. And here you have the domain growing, and then there's a kind of fingering instability where you have a branch forming. So in summary, what you have here is coarsening and anti-coarsening or domain splitting. Put in other ways, you have forward topological transitions and backward topological transitions that then maintain overall this foam-like structure. Where do we go from there? Well, I think protein systems are just beautiful, and you can create lots of systems that not just have this foamy structure, but cell-like properties. Here there's different examples. So that's an example from MIT, from Nikta Fakhri's group, where they looked at starfish oocyte. You see clearly that the patterns that you have a linking between the shape and the patterns that you have. And here you have this vesicle that moves. So I think there's a new frontier, and this new frontier is to study the interplay between protein dynamics and the dynamics of membrane. Since the chairman is getting up, I do make a little promotion here for my student. So what I think is that if you just combine proteins and membranes, you can create a lot of cell-like function by using the fact that you have chemomechanical coupling that allows you to perform mechanical work, and bulk boundary coupling that allows you, from the protein perspective, to sense the geometry. And Tom Burkhardt, who is in the audience, I hope, will present this in his work later on this day, so stay tuned. You have to stay, unfortunately, to 5 o'clock, but it's worth it. Please come to his talk. Now, let me conclude with a few words. I hope I showed you that biology has a lot of phenomena that are not only interesting from a biological perspective, but offer a very beautiful view on new physics. I told you about time complexity of self-assembly that allows you to design fast and efficient processes. And I showed you that protein patterns can form in a range from in vivo to in vitro, and actually even to a point where you can have cell-like motion. The future looks bright. There's a lot of things to perform research on, from dynamic manifolds to lifelike systems, or even to the evolutionary design of complex systems. And this slide is very important to me because it emphasizes that nothing in life you can actually achieve as an individual. It's always about the team. And the team is the beautiful students that I had a pleasure to work with over the many years. And here are a few of them that have been contributing to all the things that I've been working on and told you about in the last 35 minutes. And last but not least, my thanks goes to the experimental colleagues. They keep us as theoreticians honest and tuned to reality, which is the experimental data. So thanks for your attention.